monomers, right? Several nucleotides come together to make the polymer that is the nucleic acid. There are two nucleic acids that are there. We have RNA and DNA. So RNA stands for ribonucleic.
I have a few network problems. I'm not opening my video.
Okay. So talking about the RNA, it stands for ribonucleic acid. DNA stands for deoxyribonucleic acid. Now the question comes along, what is a nucleotide? So see, nucleotide is a combination of three things. What are the three things? Pentose sugar, everyone knows, a 5-carbon sugar. A 5-carbon sugar along with nitrogenous base and a phosphate group. These three things together, they make one nucleotide. And when several nucleotides are present, they form the polynucleotide. So first we'll talk about pentose sugar. So everyone already knows that a pentose sugar is a 5-carbon sugar. This is the sugar that has a 5-carbon cyclic structure, wherein in the case of ribose sugar, you have the OH group at the 2nd carbon position. And in the deoxyribose group, you do not have the OH group at the 2' carbon of the cycle. Isn't it, students?
Would they ask us to draw this?
Yeah, this is very good. The rest of the structures, no one will ever ask you to draw, but this too is the basic thing. Just like how for writing long sentences, we know ABCD, so this is the ABC. This too you have, but this is not ABCD, it is CHO, CHO, CHO. Okay, so this is clear. Pentose sugar is clear to everyone. This is the first component. Then the next is nitrogenous bases. Now see, nitrogenous bases, have you heard about adenine, guanine, these things before? No. See, nitrogenous bases, these are those compounds that are of two types. These are cyclic compounds, okay? Purines and pyrimidines. Purines are those compounds that have a single cyclic group. Pyrimidines usually have double cyclic, heterocyclic group. So purines, the examples for purines would be adenine and guanine. Adenine is written as A and guanine is written as G. On the other hand, there are pyrimidines like thymine, uracil, and cytosine. Okay, now see, one very important thing that people have to remember, that this thymine is the pyrimidine or the nitrogenous base that is found only in the DNA. Uracil is found only in the RNA. DNA doesn't have uracil and RNA doesn't have thymine. Now, furthermore, the third component is the phosphate group. That is, it comes from the phosphoric acid, that is H3PO4. So when all of these things are combined together, what we have is a nucleotide, and then several nucleotides come together to form the nucleic acid. The nucleoside, it is made up of two things. Now see, if you remove the phosphate group from the nucleotide, what you're left with is a nucleoside. So pentose sugar plus nitrogenous base is equal to nucleoside. When you add a phosphate group to nucleoside, you'll get a nucleotide. Is it clear? Side, tide, the difference between them is clear? Now, in the nucleoside, the bond that is formed between the pentose sugar and the nitrogenous base is the glycosidic linkage. So, whenever the nucleoside combines with the phosphate group, the bond that is there is called as an ester bond, and we get a nucleotide. So, this is the nitrogenous base along with its nucleoside and nucleotide. For example, if the nitrogenous base adenine is there, just adenine. Now, when it will combine with a sugar, we'll have adenosine. If this adenosine combines with the phosphate group, we'll have adenylic acid. Same way, when guanine combines with a sugar, we'll have guanosine, which is a nucleoside. When this guanosine will have a phosphate group attached to it, it will form guanylic acid. You people are getting me?
Yes, ma'am.
Then third, we have cytosine. So when cytosine will have a 5-carbon sugar attached, it will form a nucleoside called as cytidine. And the nucleotide associated will be cytidyllic acid. Then we have thymine, which will, as a nucleoside, it will be called as a thymidine. And as a nucleotide, it will be called as thymidyllic acid. Dear students?
Yes, ma'am.
Lastly, uracil, which is only found in the RNA. So that as a nucleoside will be called as uridine, and as a nucleotide will be called as uridylic acid. So this section was the section for the nucleotide, and the polymer of nucleotide is a nucleic acid. The example for which are DNA and RNA. Clear to everyone?
Mom, what's an ester bond?
Ester bond is the bond that is formed between the nucleoside and the phosphate group. Wait, I'll show you. One second. See this thing. The bond that is formed, that is this one here. You see how ROCH, that is your ester bond. And this is the bond that is formed in the this thing, nucleotide. Okay, so when we talk about ester bond in nucleotide, so if you see over here, look at this. See, right here, the bond between the pentose sugar, which is connected with as a nucleoside. So the bond that will be formed between the nucleoside and the phosphate group will be your ester bond. Clear? And when same, just like that, when several nucleotides will be joined together or two nucleotides will be joined together, so the bond that will be there will be phosphodiester bond. Clear?
Yes, Mom, thank you.
Now we have enzymes. See, students, enzymes, they are called as biocatalysts. What are catalysts, students? You might have studied about catalysts in chemistry.
Are those the ones that help to make the reaction go faster?
Catalysts are those substances that increase the rate of reaction, right? It forms product quickly. So when we talk about the catalysts that are found inside the living organisms, we call them as biocatalysts. The example for which is enzymes. So remember that almost 99.9% of the enzymes that are there, they are proteinaceous in nature, that I've already told you about, isn't it? Now, apart from all of this thing, you have to remember that enzymes, since they are proteinaceous structure, so they will have, so like you can say, an optimum, you can say, structure at which they are active, optimum pH at which they are active. So for that, for enzymes, you have tertiary structure, which is the most active stage of the enzyme. Now, why is tertiary structure better? If you people remember the structure of proteins, you people might remember how I told you that tertiary structure is very essential for the activity of enzyme. Why? Because since there are foldings amongst themselves, so that creates active sites. See, these curved areas, they are called as active sites. So normally, how does the enzymatic reaction happen? This is the substrate on which you have to apply the enzyme to make it turn into a product. This is a product, this is a substrate. Okay. This right here is the enzyme. So the enzyme comes, or sorry, the substrate comes and binds with the active site of the enzyme, and then this gets converted into the product. Clear? This is the basic, you can say, the thing that happens when we talk about the enzyme activity. So mostly the enzymes, they are proteinaceous in nature. Now, apart from that, there are some nucleic acids that also act like enzymes. We call them as ribozymes. Like some RNA, they can act like enzymes. So we call them as RNA enzymes, and those are like the examples for which are ribozyme, ribonuclease. Okay, is it clear, students?
Yes, ma'am.
So how does it happen? The substrate combines with the enzyme. There's a complex formed, like that's like a intermediate, very short temporary complex that is formed. It is called as the substrate enzyme complex. And then the substrate gets converted into the product and the enzyme is unused. So the thing about enzymes is that only that the whole entire reaction will happen and the enzyme will not be consumed during the chemical reaction. Clear? Meaning it will remain the way that it is. It will cause several thousands and thousands of reactions to take place. Understood? So that's the case. Now, talking about the active site. So in the enzyme, there are crevices, there are pockets like curvy structures present that we call as the active site. Now this is the site of the enzyme where a substrate would come and fit over. Now for example, every single substrate has their unique enzyme which works on them. For example, if the substrate is like this, the enzyme is going to be in a way in which it will fit here. So that the substrate and enzyme, it's like a fit like this. Every enzyme is specific for every substrate. It's not that you'll put any enzyme and the reaction will happen. No. If this is the enzyme, so the substrate also has to be in a complementary manner so that they can combine and cause the chemical reaction to take place. Clear? So every enzyme has a specific active site for which is very specific for the kind of substrate it is meant for. Now, many inorganic catalysts, they are used in laboratories for chemical reaction, but those are inorganic, something that is not found in the living organism. Whereas the other ones, the ones that we are talking about, those enzymes, they are biological catalysts or organic catalysts. Clear? Now let us see the difference in between both of them, enzymes and inorganic catalysts. So remember that almost all the enzymes, they are proteinaceous and they have a complex structure. Whereas when we talk about inorganic catalysts, they're not going to be protein. Okay, they're not proteins, rather they are like inorganic molecules like nickel, platinum. These things are added to the reaction so as to increase the rate of the reaction. Now enzymes, since they are biomolecules, they are biological molecules, so they are found inside the living organisms. On the other hand, the inorganic catalysts, they do not occur in the living cells, they are something present outside. An enzyme catalyzes only a specific reaction. It is very, very specific to the type of substrate, to the type of reaction, to temperature and everything. Whereas when we talk about inorganic catalysts, these are the ones that do not need any kind of, you can say, specificity. It can be used for several reactions. Nickel, cadmium, platinum, magnesium, these are all your inorganic catalysts. Then now the thing about enzyme is that since it is a proteinaceous structure, that too a tertiary structure, a 3D structure. So if there's like an optimum temperature at which that enzyme would work. If you increase the temperature beyond that, the enzyme is going to get denatured. If you decrease the temperature beyond that, the enzyme is going to get inactive. But these inorganic catalysts, they can work at any temperature, they can work at any pressure, any pH, no specificity. But the enzymes, they are highly efficient, meaning the reactions that you're using it for, it will cause that reaction 100%. Whereas inorganic catalysts, they are not so efficient. Clear? Now, enzymes, they work at a constant or an optimum pH, concentration and temperature. If, for example, you are increasing the temperature more than the optimum temperature, the enzyme will get denatured or it will get damaged. And the meaning of denatured is that it will lose their tertiary structure and go to their primary structure. So the enzyme is destroyed. Understood? If you decrease the temperature less than the optimum temperature, like for example, if there is an enzyme that works at 32 degrees Celsius. So if you provide 20 degrees Celsius, 28 degrees Celsius, the enzyme will get inactive. If it works at 32 degrees Celsius and you provide it 40 degrees Celsius, it will get denatured. Understood? But one exception is there. There is one enzyme that can even work at 90 degrees Celsius. Imagine, that's the special case. So there is an enzyme called as Taq polymerase. This enzyme is from 80 to 90 degrees Celsius. Now this enzyme, it is extracted from the bacteria called as Thermus aquaticus. So the word Taq, the T stands for thermus and AQ stands for aquaticus. Clear to everyone?
Yes, ma'am.
Ma'am, is this
Uh-huh.
thermoacidophile?
Hmm?
Is this a type of thermoacidophile?
Yes, it is. Also, for those of you whose internet connection is perfectly fine, please switch on your videos and show me your beautiful faces. Moving forward, talking about the nature of the enzyme action. See, the thing that I was talking to you about. See, here is the substrate and the substrate has a specific shape. Now, when this substrate will go, it will bind to that area. For example, the substrate is like this. So, the enzyme is like this, wherein the substrate would come and fit properly. Okay. And this, you can say, after this is the complex that I was talking about, enzyme and substrate complex. Now further, this enzyme will cause the reaction to take place. It will form the products and enzyme will be unused. It can be used again and again and again and it will never get efficient. Understood? So the substrate binds to the active site of the enzymes. The binding causes the change in the enzyme and the enzyme fits very tightly around the substrate. Now, the active sites that are there in the enzyme, it starts to break the chemical bonds, right? And then starts to form a different product from the substrate, right? And then the enzyme releases the product and the enzyme is ready to be used for another reaction. So enzyme action is clear to you because they can ask you this thing in exams. Like write the enzyme action. How does the enzyme work? Right? And then how is the enzyme specific? So these things you have to take. Clear?
Ma'am, can you explain it once more?
Enzyme action. This is enzyme action. Okay. So there is a substrate which you have to convert into a product. So for this substrate, a specific enzyme will be present. You will use that enzyme. So this, like now we are seeing the behind the scenes, right? The chemical scenes that are happening. So if the substrate is in this manner, it is like this. So the enzyme will be in such a way that the substrate will come and get fixed properly. Okay. This complex that is formed, it is called as the enzyme substrate complex. Now, during this is a very short time period. This is something that we can't see. We only see, we put the substrate, we put the enzyme, we mix it together and we find the product. But what goes behind it is what we're studying right now. So the enzyme substrate complex, during this time period, which is a temporary time period, there will be certain chemical reactions taking place between the substrate and the enzymes, which will, like the enzyme will change the shape of the substrate, change the properties of the substrate and create it into a product. And after this is done, the enzyme will release the product and the enzyme is left as it is, ready to use for the next. Clear?
Yeah, mom. Thank you.
Is it clear to everyone?
Yes, ma'am.
Very good. Now talking about the factors that affect enzyme activity, extremely important from, you can say from this practical point of view, right? When you people will go into higher classes, you will have to work in a lab. So you will have to know that the enzyme that you're working with, is that enzyme at the right temperature? Is it not? You know, there are a lot of enzymes that if we want to keep them safe, we keep it in an ice box so that it doesn't degrade because of the room temperature. So that you can say sensitive the enzymes are. So there are three factors that affect the enzyme activity: temperature, pH, and substrate concentration. First is temperature. This is the graph that shows you as the temperature is increasing on this x-axis, the enzyme activity increases only up till a certain point and then it starts decreasing because the enzyme is getting denatured as the temperature is getting increased. So enzymes, they generally function in a narrow range of temperature. Temperature at which the enzyme shows maximum activity is called as optimum temperature, and it is different for different enzymes. Few enzymes have 20 degree as their optimum temperature, few have 30 degree, and it varies. Okay, so you know that if you put the temperature less than the optimum temperature, the enzyme is going to get denatured, inactive. And if you increase the temperature, the enzyme is going to get denatured. So now, like for example, if either of the two things are happening, now if you put the right temperature for that enzyme again, so what will happen? The enzyme will go back to its normal state. Clear?
Yeah, mom.
Don't push her like that.
He's a boy.
Acha, don't push him like that. He wants to attend the class, no?
He was annoying me since the beginning. Mom.
Hello. All right. So now talking about the pH. So now again, the enzyme will, like most of the enzyme, majority of the enzymes, they work at a neutral pH. What is neutral pH, students? That is pH 7, isn't it? 7 pH. Higher than 7 is basic, lower than 7 is acidic, right? That's the case. So when we talk about the pH, if you decrease it, then the optimum pH, that will be acidic, and if you increase it, that will be basic or alkaline. Shouldn't be alkalis, should be alkaline. Clear? That's the case. So again, the graph would be the same as the pH increases or the pH changes, the enzyme activity at the optimum pH increases. But when you increase it furthermore, then the enzyme activity will start decreasing. So this is the second. The last thing is the substrate concentration, meaning the amount of substrate that you're going to turn into the product. If you increase that substrate, the velocity or the speed of the enzyme activity will also increase. But it will reach a maximum velocity, and beyond that, even if you increase a lot of substrate, no change will take place. Okay, so the point at which the velocity of the enzyme reaches half its value, like for example, if here the maximum velocity of the enzyme was something, but when we talk about when the maximum velocity, when it reaches half its value, that point is called as Michaelis constant, Km. Okay, it is the concentration at which the velocity of the reaction reaches half of its value. Clear? So
Yes,
for
ma'am.
example, see this thing. If at this point, the velocity is at its maximum peak. Now, even if you're increasing substrate as much as you want, the reaction is going to go this way. This way means constant. It's not going to change anymore. The velocity of the enzyme activity, the speed will not change. Clear, students?
Yes, ma'am.
Yeah, ma'am.
Now, just like how you people have studied the classification and nomenclature of organisms, same thing you have to do here also for enzymes. Okay, there are different classes of enzymes present and they are named accordingly. Okay, so when we talk about this thing, so the classes of enzymes, they are oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. These are the different classes of enzymes. Like for example, insecta, right? Insecta is a class. Under that you have mosquitoes, you have honeybees, all of that, right? So that class, so same way enzymes also have classes of enzymes under which different enzymes would come. The mnemonic to remember this is OTHLIG. Okay, I used to remember it like this. Oxidoreductase, transferase, hydrolase, lyase, isomerase, ligase. Okay, these are your various classes. Now, let us talk about them. This much you people have understood, na?
Yeah, ma'am.
Yes, ma'am.
Talking about oxidoreductase that are also called as dehydrogenase. Now, oxidoreductase. You see how two words are there. Oxidation is also there and reduction is also there. So, these are that class of enzyme that would do the oxidation reduction reaction everywhere. So, what are the enzymes that belong to this class? We have reductase enzyme, we have catalase enzyme, dehydrogenase, peroxidase. These are different enzymes that are there that will do oxidation reduction reaction. So, for example, if there is a substrate that you people are using to make a product, so you want to create an oxidation product of that substrate, the enzyme that you're going to use is this. Understood? Accordingly. Second, you have transferase. Now, these are the enzymes responsible for transferring of a specific functional group or specific group from one substrate to another. Transferase transfers something, right? So, the examples are kinase, transaminase. Then, and see, one more thing I would like to tell you that these classes of enzymes and their examples are very important. So I say the whole entire chapter is important. But to pinpoint the specific things that you shouldn't skip, because I know when you people study, you choose your favorite topics and you study those only. And you're like, the rest, I'll not attend, I'll not do. So no, this is the part that even if you don't want to study, you have to. Understood? Because the examples of these classes and the name of the classes, they usually come like, for example, they'll give you in MCQ that this enzyme, like transaminase, belongs to which of the following class of enzymes. So it belongs to transferase, like that. Or they'll ask you to match the following, or they'll ask you these things in assertion reason. So you people should know. That's the bottom line. And I want full marks from everyone. No concession over there. No bargaining. Full means full, right? Everyone has to prepare. And yes, one more thing, students, if anyone is having any trouble in learning something, in understanding class 11th bio, you can talk to me about it freely. Why? Because at the end of the day, I want the perfect result in biology. That's my responsibility for you guys. So I want you all to study, to learn. Like you have few months only, right? And then inshallah, you people will be promoted to class 12. So before you guys be promoted to class 12, I want you all to give, or you can say dedicate yourself for those few months for your finals and do as I say. Follow me. Understood? And see, no one will keep you away from full marks. No one. No strict teacher, no heavy checking teacher, no. No one will keep you away from full marks if you follow what I say. Understood? The notes that I've provided to you, they are more than enough. Trust me. Like NCRT, you take out a chapter, for example, you're studying something. You take out my notes first, you read my notes thoroughly. Then you go to NCRT. That's more than enough at this point. More than enough. You won't need any kind of reference book, nothing you will need. What I am giving you in my notes, what you people copy down in the class or whatever notes I provide you, that's sufficient for your examination. Understood? When you know every single thing of any topic, then you can solve any question, isn't it? When are we not able to solve any question? When our concept is not clear, isn't it? But what if our concept is completely clear? Now anyone would come and ask you anything and you'll be able to answer. Getting my point, students? So no matter whatever happens, before your examination, these few months, you dedicate yourself to your subject. I mean, I'm talking about myself. Okay. So you dedicate yourself in such a way that none of you score below 90. That's our aim. Clear to everyone?
Yes, ma'am.
Yeah, ma'am.
And you people can do it because I've seen all of yours performances in the tests over here. And I know none of you cheat. And I see how you write your answers and it's flawless. To be very honest, it's very nice. So you keep up that hard work or whatever it is that you people are doing. For some of you, it will be really hard work. For some of you, it would be a piece of cake. It can happen. Everyone has a different intellect. It can happen. So try to give in your best so that at the end of the day, you will know that, okay, I gave my best, I did my best. And you'll be very satisfied with your answer. And you people know now, once you people are done with your finals, I ask your exam marks also. I'm like
Yes.
sitting here waiting for your report cards. Okay. So inshallah, I'm really hoping the best for you all. So if you prepare it nicely, no one is stopping you from anything. And one more pro I'll tell you. If you learn your 11th class right now, only because in your finals you'll get all your chapters, isn't it? The whole entire portion you'll have now. If you learn it by heart and you give it all right now, so for those of you, for those of you who are planning to take that NEET examination, you don't even have to work that hard post your 12th. Imagine. Just imagine, like once you're done with your 12th and you try to apply for your NEET examination, you won't even have to work that hard. So mark my words, start your, even if you're not studying, I don't care. From today onwards, you make it a habit that every day I will give 1 hour to biology. Every day. And one concession I'll do, for those days you have classes with me, that day you leave that hour. That day 1.5 hour is mine. But every day you people do 1 hour biology, no matter whichever chapter you want. Understood? Clear? If you want to start with human physio, all of you love human physiology only. So you start with that. I'm fine with it also. But whatever you do, make it in such a way that no matter whatever question any teacher comes and asks, not just me, anyone would come and ask and you people will be able to answer. Got it?
Yes, ma'am.
So
Yes,
enough
ma'am.
bhashan for today. Let's continue. So hydrolase. This is the third class of enzyme. Now hydro, water, right? And these are those enzymes that will break down big molecules into smaller molecules in the presence of water. Hydrolase. Okay. Example, all the digestive enzymes like protease, amylase, maltase. These are all your digestive enzymes that are in nature hydrolase or hydrolytic enzymes. Understood?
Yes, ma'am.
These are hydro. Furthermore, we have lyase. Now, these enzymes, they catalyze the cleavage. What is cleavage? Cleavage means breakdown of substrate into two parts. Okay. A double bond is formed at the place of removal of the group. Okay. And this is very different from hydrolysis because here water is not involved. Breakdown is happening, no water is involved, and wherever a part is removed there, a double bond is formed. That is ligase. For example, aldolase enzyme, decarboxylase enzyme, there's carbonic anhydrase as well. You people remember carbonic anhydrase? Where have you heard this before? Where have you heard this before? No one has heard this before. And this is the first time I'm listening to this. Acha, breathing and respiration.
It's an enzyme, right?
Hi, it is an enzyme. I've given you the name of the chapter, breathing chapter. Now, can you remember carbonic anhydrase?
A catalyst.
Huh?
Catalyzes.
It's a catalyst to make that HCO2.
You knew. Yes, bicarbonate ion formation. You people remember?
Yeah.
How CO2 is, transferred in the form of bicarbonate ion. So for that, the enzyme that was there was carbonic anhydrase. Understood? Then we have isomerase. Now these are the enzyme, or this class of enzyme. Basically, it creates isomers. Okay, it rearranges the molecules and forms isomers. What are isomers? These are those molecules that have same molecular formula, but they have different structural orientation. The example is isomerase. This is ligase. Now, ligase is like a glue. Okay, it joins two things together. It joins two molecules, two substrates together by forming covalent bonding, meaning sharing of electrons is done, right? Like CO, CS, CN, these all bonds, they utilize, these are the products of ligation. And here ATP is utilized for joining. So the examples are RUBP carboxylase, Rubisco enzyme that acts like a carboxylase, then ligase enzyme, then phosphoenol pyruvate carboxylase. These are all the enzymes that are ligase, that belongs to the class ligase. Clear, students? Now, talking about properties of enzymes. So enzymes, they are proteinaceous in nature. I think the chapter is now about to be over. Little bit only. So the enzymes, they are proteinaceous in nature. They increase the rate of chemical reaction. They are highly efficient, good for what they are supposed to work. They are unchanged and unused after every reaction. And they are highly specific. Specific for a type of substrate. Now, this was the main enzyme portion. Now, enzymes, they require other molecules or other substances to enhance their activity. And those stuff that enhances the enzyme's activity, we call them as cofactors. Co means together, and the factor that enhances the activity of the enzyme. So for example, if you talk about a holoenzyme means a complete enzyme that has a protein part called as the apoenzyme, combines with the cofactor, and then only you get a completely working holoenzyme, a complete enzyme. Now these supportive agents that support the enzyme, they are of three types: prosthetic group, coenzymes, and metal ions. Talking about prosthetic groups, these are organic compounds that bind very tightly to the apoenzyme and increase its efficiency. So that is prosthetic group. Okay, it is organic. This is important to remember. It is organic compound. Second, we have coenzyme. Now, these are also organic, but their association with the enzyme is for a short period. And here you see it's for a tight association for a long period, short association for a very tiny amount of period. So the essential component of many coenzymes are vitamins. For example, the coenzyme NAD, right? You know, nicotinamide adenine dinucleotide, all of you know now. So this contains the vitamin niacin in it, which increases its efficiency. Lastly, you have the metal ion. So these are not organic, and these are inorganic substances that increase or enhance the rate of chemical reaction. So many enzymes, they require a lot of metal ions to work. So for example, if we talk about like the enzyme called as carboxypeptidase, this enzyme works with zinc. So when you apply carboxypeptidase in a reaction and you add zinc, then the reaction will go very quickly and form you the product. So catalytic activity is lost when the cofactor is removed from the enzyme. So this tells us that for the catalytic role of the enzyme, for the superior catalysis by the enzyme, cofactors are very important. And all in all, what are cofactors? These are those factors, these are those molecules, those chemicals that enhances the activity of enzyme. Clear?
Yes, ma'am.
Now there is this particular graph that shows you how if the enzyme is not present for a chemical reaction, the amount of energy required to complete the rate of reaction or to form the product will be very high. But when you apply the enzyme to the chemical reaction, the activation energy reduces. Clear? Okay. So now you people write a little bit. A few minutes, then I'm starting a new chapter. I'll send these notes. So what is the last thing you people have written? Tell me. Hmm?
We have to start from disaccharides.
Dissect.
Yeah, from there, amongst the disaccharides.
Which one? Amongst the disaccharide.
Yeah.
Done now.
Done, students?
Yes, ma'am.
Done, students? Shall I scroll down?
Yes, ma'am.
Done, students. Shall I scroll down?
Yes, ma'am.
Yeah, ma'am.
Done, students? Shall I scroll down?
Yes, ma'am.
All right, students, let's